Hi, my name is Nadine Ziemert. I'm a professor at the University of Tübingen for microbiology and bioinformatics, and we are interested in secondary metabolites of bacteria. Secondary metabolites are chemical compounds that are not primary metabolites. That means they're not necessarily used for growth or for direct survival, but they have really specific functions in bacteria. And we think they're really interesting because we use them a lot as medicine. For example, almost all our antibiotics are secondary metabolites produced by bacteria. Anti-cancer compounds are produced by bacteria. We are not necessarily only interested in these compounds because they're used in medicine, but for these bacteria, they're really fundamental units with which they sense and respond to their environment. For example, they use this as signaling molecules, as weapons against other bacteria. So they really have a huge effect of the bacterial or microbial diversity out there. And we know now that our microbiome or the soil microbiome or plant microbiome have huge effects on the health of their hosts. So this really means that these chemical molecules really have a big impact on the health of the earth, actually. Our research question is, what kind of chemistry bacteria produce? How much diversity is out there in these chemicals that these bacteria produce? And what is their function and their impact on the microbial communities and on the host? So we are interested, for example, in who exactly produces what kind of chemical compounds. We're interested in how this diversity is evolving and where can we find most chemical compounds, for example, that are most promising as drugs in medicine, as antibiotics, for example. Some specific research questions that we have is, for example, what kind of bacteria produce the most promising antibiotics? Where can we find them? What kind of chemistry is out there in the environment? If we, for example, map kind of the whole world of genes that encode for this chemistry so that we can find them more easily in the future. And we are interested specifically in how these diverse molecules evolve. So how does nature evolve into this, or how does nature evolve these molecules? And to find now this huge diversity of molecules out there in nature. We're a very interdisciplinary lab. We work together with bioinformaticians, biochemists, and microbiologists. And we're using first computational methods that we also develop new to prioritize gene clusters, predict certain compounds, and then we go in the lab and try to clone them, heterologously express them, or use synthetic biology to actually find the compounds. And by then going back to the computer scientist, we can have an iterative process where we then can improve our methods in computational biology, make predictions again, check these predictions in the wet lab, and then go back to the computational biology and improve our methods even further. We developed a compendium of bacteria that are the most promising producers of secondary metabolites and that haven't been studied so far. So we kind of built a roadmap where we can predict where we think it's most efficient to look for new antibiotics, for example. And another finding that we have that I think is kind of my favorite story at the moment is where we are looking at specific compound families and how they evolve. So we can use phylogenetic methods and make phylogenetic trees and can see how certain compounds evolved into different specific chemistry, into different mode of actions. And we did that, for example, for a very well-known group of antibiotics, the glycopeptides. And we were able to understand the evolutionary history and the diversification of these glycopeptides. And at the same time, we're able to go back in time and predict computationally how the precursor of and the ancestor of these glycopeptide molecules looked like, that we called paleomycene. So we estimate it's about a couple of million years old. And we were then able to go in the lab use synthetic biology to make the compound and even show that it was already active, probably with the same mode of action like the known glycopeptides that we have today. So why do I think my research is relevant? We have the problem of antibiotic-resistant bacteria, so more and more antibiotics are not working anymore, and we know that this problem will be even worse in the future. So what we urgently need is novel antibiotics, so our research helps with that. We find roadmaps and tools, or we develop tools that helps us to make drug discovery more efficient by, for example, telling where can we find them in nature and what kind of bacteria. But also we develop tools to prioritize certain gene clusters that we think encode for antibiotics with novel mode of actions, that we can then find them more easily by synthetic biology methods, for example. But also we're interested in understanding the function of these specific compounds within microbial communities and using then these molecules to make microbial communities, for example, more resilient against pathogens, so we don't need specific antibiotics. So all of that kind of research, I think, will help in the future to fight infections. That's a goal of our cluster, CMFI, controlling microbes to fight infections. And I think that will generally also help to understand microbial communities and how they infect or affect us. My research vision for the future is clear that I think it will be more and more computational biology. We will use more data, we will create more data and be able then to faster make predictions, develop new molecules and really test them easily and fast in computational methods, and then only really go into the lab with very specific things. So my goal is improving structure prediction, that we can say, okay, we have a gene cluster here, so in the future we can predict exactly what kind of chemistry is encoded within that gene cluster, and then we can go further and check what kind of and predict what kind of bioactivity this structure will have, and then already predict in the computation or within computational methods if that will be a good antibiotic or not, and then really only go forward with the most promising candidates to find novel antibiotics, for example. And in general, I think also really modeling communities, modeling what the function of these molecules within communities will be a big part of it, and understanding then when I add a certain molecule into a microbial community, how can I edit this microbial community, make it, for example, more healthy or more useful for certain hosts like plants or humans, but also how can I stabilize these communities to make them more resilient against climate change. So I think that a lot of these methods and developments in the future can help to maintain diversity.